So notice, the amino acid's down here, the code is up there, there's a transfer RNA in between. This guy never touches that code. But the transfer RNA knows exactly what to do. And by the way, every transfer RNA that does that has to have a specific protein to make that happen. How many amino acids are there, class? Do you remember?
20.
20. So there's 20 specific proteins for those 20 different amino acids to make that happen. All right, so far we have a protein that has one amino acid in it. One. You know what a typical protein has in it? A string, how long, you think? Typical protein in your body. Take a guess. 50. How about 1,000? You listening to me? A typical protein has 1,000 amino acids in a row. And you know how long it takes to build it? Microseconds. But notice what happens. This is way too slow. But watch. This guy gets finished. The next one comes up. Go ahead. This is the next transfer RNA. He combines with that code. What? Stop right there. Now, that's got to go away. But you, look, you see this one is now hooked up to that one, which is now hooked up to that one. They form chemical bonds down here. But how did you get those three from the transfer RNAs that can match exactly those codes? So now you have 3 amino acids in a row based on the code that was in the DNA.
Yeah.
All right, let's continue. So now you got a 3-strand protein. Here comes the 4th one. Here comes the 5th one. Here comes the 6th one. Speed it up, you guys. This is ridiculous. Come on, do it. Whoosh, whoosh, come on. It's way faster than that. They're trying to show you how fast that is. Stop. I don't know how many we've got now. But you know how many a typical one is? 1,000. That same process. 1,000 times. I'm tired. And your body, ladies and gentlemen, while you've been sitting here watching me, has been building proteins like that at a phenomenal rate. You wonder why you get tired just sitting? Your cells are busy. They're doing it all the time.
And you're standing and doing it.
I know. And I'm old. All right, keep going. Isn't that fascinating? All right, stop. Now, where are we here? This is the messenger RNA. You notice it's going through and coming back out?
Yeah.
And you see down here, it's hard to see. There's a little blob coming out down here. That's the protein that was just built.
Okay.
And it comes out the bottom. All right, let's keep going. Be careful to stop when I say, okay? So there's the protein. See it right there? Oh, you stopped it, didn't you? Okay. I didn't say stop it, you did, but that's okay. Go ahead. All right, stop. Okay. Now, you see the protein's been released from the ribosome. And it's formed already in kind of a shape. It's not just a long strand. And here's something, this is my 5th really important thing. Proteins do not work if they're just a long strand. They don't work. They have to fold up to it. So there's a primary structure, and that's the string of amino acids. But then they fold up into a secondary structure. And then they fold again into a tertiary structure, and until all that happens, you don't have a functioning protein. All right? Very important. Keep going. So we've got our secondary structure going now, right here. It's taken over to another organelle inside the cell that closes in on it, and look, just watch it. And of course, it doesn't look just like that. But notice, see how it's twisting and turning around and forming itself into a specific shape and folding on itself until that folding, you see how that rotated? Until it's complete and it turns white. Wrong. Stop. Stop. It doesn't turn white. But the turning white means it's finished. and then it's released out into the cell to do the job for which it was designed. And I want to tell you, ladies and gentlemen of the jury, if that isn't designed, I want to help you understand how Henry Ford builds cars. Henry Ford's factory is, don't touch top side or bottom of this. Microsoft's computer factories do not touch top side or bottom of this. And yet people will sit around and say, isn't it fascinating how nature produced that? I don't believe it. It is beyond belief. And there's way more to the story than we've even talked about. Go ahead. So it's being released out into the cytoplasm to go do the job for which it was designed. Now, stop right there. This book by Douglas Axe is called Undeniable. Very recent copyright date. Douglas Axe was doing research on the folding of proteins in the early 2000s. And one of the things he wanted to discover is, can it fold into just lots of different forms? And what are the restrictions on how a protein folds up? And without going into the massive detail, which you can read in here, it is a very limited way that that protein can fold that even works. Anything else you do doesn't work. And that's just the folding piece. And he demonstrated that scientifically by repeatable experiment. And you know the reaction? Embarrassment shut this guy up. Because it doesn't fit with the common worldview. But it's powerfully solid science.
What was his name?
Douglas Axe, A-X-E. Read after him. He's outstanding. Okay, that's enough on that. Let's get back to the slides if we could. Okay. So you can toggle back now? Okay. There we go. All right. Class, I'm wearing you out, I know. But isn't this exciting?
Yes.
Utterly. So we've talked about transcription, migration, and translation, haven't we, for protein building? Every one of those steps takes
Protein.
Proteins.
plus the DNA. DNA without proteins is useless. Proteins without DNA is also useless. Now, a little more information that we've learned since. You see across the top there, that's the DNA. Do you see it unzipped there?
Yeah.
And you see it making the copy of the RNA. And here's the transfer RNA and the ribosomal RNA. That's two things I haven't even talked about tonight. See, there's a bunch of other stuff going on too. We haven't talked about. The messenger RNA, do you see it's got those little white spots in it? Those are called introns. We didn't know anything about introns until the 1980s and 1990s. And we began to see that when that copy was made, that whole thing was not copied and translated. It had to be cleaned up first. Listen. The introns were snipped out. You see how it's reorganized there? That's called a mature messenger RNA. That information started coming to light. It was a great discovery. It was amazing that some of the pieces of that copy were not used. They were introns. The exons were used, but not the introns. So we snipped those out and reorganized it into a mature messenger RNA before it migrated. Guess what it takes to do that?
Proteins.
Exactly right. They're called spliceosomes and editosomes. Specific proteins that splice it and sew it back together. Okay? Wonder what people who don't believe in design said about that. My other lawyer friend here. who would be presenting his case for nature, he would say, why would some designer build a system where you have to copy it and then you got to edit it before you can use it? That's a stupid system. That doesn't look like design to me. So why do you say this is design? And the more we learn, the less it looks like design is what they would say. And they thought the introns were just junk that were thrown away. Interesting. And I will tell you, as a believer that this is intelligently designed, this threw me for a loop, because I didn't know how to answer it. And I don't have time tonight, class, to tell you the amazing story in the last 20 years of what we learned about introns and the whole system. It's more complex than anybody ever dreamed. It's 10 million times more complicated than what I've told you tonight. So here's what I'm going to do. I'm going to go through this so fast your eyes are going to blink. Just want you to see, I'm not out of date here. The marvelous living cell is a marvel of detailed and complex architecture seen through a microscope. There's an appearance of almost frenetic activity. Is that an English word you know? Frenetic.
Yep.
May I illustrate it? That's frenetic. Okay? That's what the cell looks like. On a deeper level, it is known that molecules are being synthesized, that is, built at an enormous rate. Almost any enzyme catalyzes the synthesis, the building, of more than 100 other molecules per second. And there are thousands of enzymes. A bacterial cell has the information content of about 100 million pages of Encyclopedia Britannica, which doesn't have any pictures. Over 200,000 molecules are being built every second. If we received a message from space containing just the genetic code and an explanation of how it was used to build proteins, there would be no one on this earth that wouldn't put it in the headlines. Somebody intelligent did that. Have you heard of the Human Genome Project? Is that what you were talking about?
My brother worked on the project.
Did it? That was in the early 2000s, and I think they finished, it started in the 90s, didn't it?
He was in the 90s.
Finished up in the early 2000s. The whole human DNA was made, right? You were alive for this. Maybe Linda wasn't, but most of us were. And what they learned, class, when they did all that mapping, was that most of that DNA was not used for building proteins. I'm pausing again. In other words, it appears that only about 5% of the DNA was needed to do all these amazing things I just told you. So what about the other 95%? You know what people started saying in the 90s and early 2000s? DNA is mostly junk. It doesn't look like design to me. It's mostly junk. So those introns, that's just junk you snip out and throw away. The other 95% of that DNA, you don't need that to make the proteins. It's just sitting around there. It's left over from your evolutionary past. Pardon me, I used the word. But that's what everybody says. Here's what I'm going to tell you. In the last 20 years, we've learned that those introns do all kinds of things, including building RNAs. Lots of small RNAs built out of those introns. They do lots of stuff. And what we thought was a detriment has become an amazing benefit. That same sequence of DNA amino acids, you remember that string that had to have snipped out of it? We have learned that that same sequence of DNA can make as many as 1,000 different proteins by zipping it and reshaping it with the spliceosomes and the editosomes. So instead of being a negative, we've learned it is an amazing positive. So you better watch out what you call junk. You might ought to be humble enough to say, I just don't know what's going on here yet. But the evidence for design has immensely improved when you look at DNA. The signatures are. So I say with Michael Behe, everywhere we look, from the macroscopic to the microscopic, things sure do look like they've been made. There's a loud, clear, piercing cry of design. And by the way, he wrote it in capital letters with 2 exclamation points. So it was okay for me to yell on that. And that's what I believe, ladies and gentlemen of the jury. This was not a natural phenomenon. This, and a whole host of other things I can tell you, was designed. That's my thought for tonight. All right. It is 5 after 8. Did it feel like an hour and a half? I hope not. All right, do you have any questions, anybody?
I noticed.
You learned about protein.
So